We're going to introduce our next presenter, Tilman Grune. He is one of our extramural researchers whose research we fund. He's the scientific director of the German Institute of Human Nutrition, Potsdam-Reichenbach, and he will be presenting on LIpofuscin, an active player in senescent cells, and how to get rid of it. Again, you can read his presentation summary and bio on the event's website and find him later at the research booth or chat with him to meet. Now let's welcome him on stage. Enjoy the presentation.
Yeah. Thank you very much. Can you hear me well now? So waiting for the presentation. Thank you very much for the introduction. I'm sitting here, as you heard now in Germany. I'm not from the Einstein College as it was shown at the beginning. I cannot see the presentation, so can you technical support somehow?
Hi, you can click the presentation button at the bottom center of your screen and it's going to open that up for you.
Ah, that one.
There you go.
Okay. Yeah, okay. Thank you very much. So I'm going to talk today about LIpofuscin or protein aggregates. So as you might know, protein aggregates are accumulating in aging cells, and this is a permanent process. In contrast to many other models we have, you will hear about that, or many other information you will get. We do know about protein aggregates already very long time. I mean, here I digged out a very old publication. As you can see, it's from 1843. So basically in the US, it was long before the Civil War. And this author here, Hannover, it was called, already presented the first drawings of cells accumulating here in the perinuclear area, protein aggregates. And basically that what was continued over the decades afterwards. Later on, around the turn of the last century, you see why are these protein aggregates formed, these age-related protein aggregates formed, and how come they are distributed in the cell. So we do know today that several types of cells in the human body contain these protein aggregates called lipofuscin. These are predominantly or exclusively post-mitotic cells. So you find them in neurons, you find it in beta cells, you find it in muscle cells, and of course here also in certain populations of fibroblasts, as you can see here. That's the fibroblast. The nucleus is essentially free of these protein aggregates. And all what you can see, that's not the staining, that's the so-called autofluorescent. You're saying it's a little bit weak here, but the same you can see for cardiomyocytes, and here on the side you see isolated lipofuscin we are talking later about. So lipofuscin is in contrast to other protein aggregates, not an amyloid-like ordered structure, like the paired helical filament you heard just about, or the A-beta structures of the brain. This is a random coiled protein aggregate, many cross-linked, which might be either due to oxidation or due to Maillard reactions here. Peroxidized lipids are cross-linking these proteins. What is important is, and that's true for all the protein aggregates accumulating with aging, they are resistant to mammalian proteases. So basically, we cannot get rid of them. Let's say not so easily, or biology has not foreseen to get rid of them. Some of them contain active transition metals. I will talk about this in a couple of minutes. Basically, why lipofuscin is formed. So that's in a cell, of course, you have a constant exposure to internal stimuli. Might be radiation, it might be oxidants from the outside, it might be metabolic stimuli like glucose, for example, or ribose, which are highly reactive as proteins, and this is damaging proteins. These damaged proteins are degraded basically, let me go back, by intracellular proteases. One of that is depicted here. That's the so-called proteasome, the core of the ubiquitin proteasomal system. But our cells, mammalian cells, let's say, are full of different proteolytic systems. The proteasomal system is there. There are caspases, which are calcium-activated proteases. Lots of different special proteases, and the lysosomal caspsins, which is a major second major proteolytic system. So basically, the proteasomal system and the lysosomal caspsin are responsible for the degradation of the bulk of intracellular proteins. If you perform an experiment in vitro about the accumulation of oxidized protein aggregates, you find something like that. As long as the cell, these are the dark columns here, as long as the cells are proliferating, they are almost not accumulating protein aggregates. So basically, we think today we have the model that if the protein aggregate is formed and the cell is splitting into daughter cells, the protein aggregates are more or less randomly distributed in the daughter cells. So you will have a permanent level of or permanent equilibrium between distribution of daughter cells and formation of LIpofuscin or protein aggregates. And the empty column here is a post-mitotic aging. As soon as you make the cells stop to divide, they start to accumulate protein aggregates. This is a fibroblast cell clone experiment here. We performed in between with many different clones of fibroblasts, showing all the same principle. And also in the older publications of the last decades, histologists and anatomy textbook describe lipofuscin in non-dividing cells. So that is basically our model that we have. You have the normal protein pool here in the middle as a cylinder, the green one. Due to different influences, these proteins unfold, are damaged, and either they are degraded, here depicted by the proteasomal system, or due to, with time, depending on the concentration of the damaging agent and of the crowding of the proteins, of many factors, for example, like chaperones in the cell, these unfolded damaged proteins form initial protein aggregates. And then you imagine these protein aggregates not as a normal protein structure as you know it from the textbook, but they are modified. There are hydroperoxides, there are aldehyde group, keto groups on the surface, and they start to cross-link. And these cross-linked initial aggregates can grow with time again. Many factors, antioxidative, oxidative processes play a role. Again, cross-linking, and the cycle will repeat many, many times. And finally, you end up with a situation where the amount of these protein aggregates is influencing the metabolism of the cell. So it was described that several neurons of elderly 80-year-old people are filled up to 70 to 80% of the cellular volume with these protein aggregates. And you can easily imagine this will influence the function of the neuron. But there's one last line of defense, and this is the so-called agrophagy, or also the autophagy, which is specialized on protein aggregates. Basically, it's a form of the macroautophagy where a membrane grows around a cellular structure, in this case a protein aggregate, and circles then in an autophagosome and then going to the lysosome. So basically, this, without going into the detail, this is a partial detoxification in the endosomal lysosomal compartment for these protein aggregates. I will show you some data on that a little bit later. So if you look for a cell which accumulated protein aggregates, in this case again a protein fibroblast, you see here in the upper right hand, these red dots, this is the lipofuscin. And you see clearly it's not evenly distributed throughout the cell. And if you stain here in green, that's the cytosolic staining. You see that there where the endosomal structures are, there are holes in the lysosome, in the cytosol. So there are basically the lipofuscin is separated from the cytosol so that it might not influence at the very beginning the cytosolic function of the cell. However, what was shown by others, Ulf Brunk and Alex Tilman, for example, that these lysosomes, if they are filled with lipofuscin, are rigid, the membrane will leak, and the protein aggregates leave the lysosome again, are spilling out in the cytosol, taken up again, so that's a permanent process back and forward. But basically, in our hands, some 90-95% of the lipofuscin at any stage is enclosed by a membrane. So we addressed the question basically 2 decades ago. We started with all that. What are the effects of these protein aggregates? How do they influence the metabolism, which was a hypothesis at the time, and we went on to study how, and basically not only the metabolism, but also, this is more recent word, also the cell function. So what we did, and that's what we started with, we produced so-called artificial lipofuscin. So what you do, you take a membrane which is rich in proteins, like the erythrocyte membrane. Some people took mitochondrial membranes. You take this mixture of proteins and lipids and damages heavily. We took a mixture of UVA, UVB, and damaged them beyond peroxidation. So it's far beyond any what normally is done, and you form some structure which we called artificial lipofuscin, and this material is in structure, in size, in the spectral characteristics, much like the natural occurring lipofuscin. So we use this stuff and expose this to, first of all, to the proteasome. The proteasome, basically, just to remember you, is responsible for degrading damaged proteins. But if there are too many damaged proteins and protein aggregates here already, as you can see in this inhibition curve, these protein aggregates block the proteasome. That is something like a circulus vitiosus, so the cells accumulating more and more protein aggregates, their possibility to degrade damaged proteins declines and declines and declines, and basically you speed up the accumulation of protein aggregates. That's a pure in vitro experiment here. We isolated the proteasome, exposed them to the protein aggregates, and looked for a secondary substrate whether it's degraded or increasingly not degraded here with increasing concentration of the aggregates. What about cells? So interestingly, and many people don't know it, if you give particles to a cell in a cell culture, in this case fibroblast, most of the cells taking up material from the outside. And in this electron micrograph here, you can see these dark structures. This is lipofuscin, artificial lipofuscin, in this case taken up by these fibroblasts. And over the weeks here in the column graph, you see that over the weeks, the autofluorescence in these fibroblasts is increasing, which is basically they take up more and more of these protein aggregates. It's a slow process, so it's not like signal transduction, which is in minutes and seconds. Here we talk about hours, days, or in this case, weeks. What you can see in the other graph, especially in the lower graph, you see that the lipofuscin-treated fibroblasts lose activity in the proteasome. It might not look like much because it's a 25% decline in the proteasomal activity, but seeing the importance of this proteolytic system for the whole metabolism, it is a tremendous change in the metabolism of these cells. At the same time, at the upper part of the graph, you have more lysosomal cathepsins, which is reflecting basically that most of the taken up lipofuscin is located in the liposomes. So basically you have more lysosomes at this time. So the question arose from us, why is it? Why is the lipofuscin blocking the proteasome? And Annika Höhn, at this time she was a PhD student, now she is a postdoc in my lab, did a very clever experiment. She took the LIpofuscin, the artificial LIpofuscin, and we imagine it as a cross-linked core with all these loosely sticking ends of peptides there, which are not totally oxidized, but still peptides. But she took a protease K, and fungi protease, which is very active, and this protease K is chewing up all these loose ends. And what stays there is the core cross-linked lipofuscin, which is not degradable even by the protease K. And she used this material and looked for the metabolic effects. And if you can see here, the first column always is a control. Then you have the effect of the lipofuscin on the total protein turnover in the cell. And then you have the third column is the lipofuscin degraded. So that's basically only the core lipofuscin. And you can see it also, it's influencing the total proteolysis, the protein turnover in the cell. It's much less than the artificial lipofuscin. The same is true for the other columns. And what you can see at the last one that brought us up, if you feed the cells with this material and then you re-isolate it from the cells, you see that to the lipofuscin, the proteasome is bond. So the proteasome as a protease is binding to the protein aggregate. And this is more important in the lipofuscin, which is a normal lipofuscin which we use, compared to the pre-degraded lipofuscin. And that is our model, how it works now. You have the lipofuscin and the upper lane, the A part here. The lipofuscin, the proteasome, is binding to it due to the loosely sticking ends and trying to degrade it. And therefore, a lot of the proteasome is out of the actual catalytic cycle of what the proteasomes are doing normally in the cell. So if you degrade these loosely sticking ends, less proteasome is binding because there is no moieties where they can bind, and therefore the inhibition of the proteasome is less important. So it would be desirable to not only to, if you can't remove this lipofuscin from a cell during aging, just to make these loosely sticking ends degraded somehow, or make them less of them, in order to have some positive effect on aging. So basically that was one of the first effects of protein aggregates. They can inhibit the proteasome, or here's the ubiquitin proteasomal system. The next effect that we found over the years was, it is known that lipofuscin contains metals. It can actively bind metals. And we did here an experiment where we included iron into this artificial lipofuscin, so you have a maximum loading here. And what is interestingly, this iron is redox active. So whenever you have, say, an oxidant or some pre-oxidant like hydrogen peroxide, it's actively forming here. You see the spectrum of the DMPO signal in the ESR. So for the iron-bound lipofuscin, so it's converting it to the hydroxyl radical, and the hydroxyl radical is a readily active damaging agent. And in some studies, we could show that in aged post-mitotic senescent cells, the lipofuscin is contributing, besides mitochondria, significantly to the oxidative stress. So that's basically the second basic biochemical mechanism that we know today of these protein aggregates. And of course, these basic mechanisms have secondary effects. One of them I will show you here. So if you block the uptake of lipofuscin into the lysosomes, that's a cell line here, the ATG5 protein is important for the macroautophagy. If you have a knockout, that's a mouse fibroblast derived from a mouse, knockout mouse here, you can block the uptake of lipofuscin here. That's the normal uptake here, and the red arrow shows in the knockout animals it's not happening. And if you look for the panel B, the first line, there's a red arrow here. It shows that you have a lot of free cytosolic lipofuscin. And this free cytosolic lipofuscin influences cell viability. As you can see here, the two dots, where we have different methods to knock out fibroblasts, as the iRNA, or also the inhibition of the acidification of the lysosome, which plays some role, but not an exclusive. So basically, the important thing here is the membrane around the lipofuscin, not the possibility of the lysosomes to degrade it, because that is anyway limited. So the next effect, and we could show that the loss in cell viability is due to apoptotic mechanisms. So the next effect is basically the apoptosis induction by these protein aggregates. And in another study here, Jose Castro, who is a Portuguese fellow in my lab, it was a postdoc, he's now back in Portugal, he basically has shown in experiments that protein aggregates, he used a special form of protein aggregates like actin aggregates in immune cells, or an immune cell model, which are Jurkat cells, and he could show that just the presence of protein aggregates blocks the cell proliferation. So it's a vividly proliferating cell line, and this cell line stops proliferating if there are protein aggregates present. So we are talking here about protein aggregates. We are not talking about oxidative damage or cell cycle arrest due to oxidative damage. It's just the presence of the protein aggregates, and we think it's due to blocking of the proteasomal system, which is important for the cell cycle. So proliferation inhibition is the next one. And for the next effect, which I want to present, I just want to present you this scheme. We came Betül Katagol, or Kademir today, she's back in Turkey, a Turkish bright PhD student working with me. We were revealed it, and we were always studying the proteasome, which is depicted in the left hand side of the figure. The proteasome is degrading oxidized proteins, and peptides are released and whatever. On the other hand, we should not forget that the proteasome is also degrading a lot of regulatory proteins. One of them, in this example, is the Jun protein, and everyone knows probably the cascade Jun is phosphorylated by junk. Then you have Jun, the phosphorylated Jun, and phosphorylated Jun is part of the AP1 transcription factor. And the AP1 transcription factor is expressing the matrix metalloproteinase 1, an important enzyme in the skin, for example, or in the connective tissue. So if you have now a lot of protein aggregates, the proteasome is blocked. And of course, as I told you before, the degradation of oxidized proteins is blocked due to a reduced activity of the proteasome. But since the proteasome is one of the limiting factors of regulating the amount of Jun, so this pre-transcription factor, if you block the proteasome, you have an accumulation of Jun, of the protein. And if you have a lot of Jun, you have a lot of phosphorylated Jun, and you have a lot of transcription factors, AP1, and a lot of MMP1, the matrix metalloproteinase. So by inhibiting the proteasome, you can increase the expression of other proteins in the cell. And that's basically what you see in skin, on UV damaged skin, UVA damaged skin, I should say. UVA is forming a lot of protein aggregates, blocking the proteasome, and you have a lot of pro-MMP1, which is leading to a process in skin we are calling photoaging. We just studied it here. In between, you have about 5 or 6 different transcription factors which are shown to be increased or modulated by the activity of protein aggregates. So transcription factor activation inhibition depending on the system you are studying. And now we are coming into an area which is more recent, and I hope it will work here. We are testing one typical post-mitotic cells, which is it's the cardiomyocytes. I'm not sure whether the video is working. No, it's not. So I'm sorry for that. What you basically should see here in the upper part of the is the cell pictogram here, and the cell is beating, so it's contracting. And you see here the contraction line, and here you see one of these contractions. You see it's going up and down, which is mimicking something like the individual heartbeat or beat of a cardiomyocyte. If you preload these cardiomyocytes, these are primary cardiomyocytes, if you preload them with lipofuscin, you have a dramatic change in the cell length, in the amplitude of the contraction, in the velocity of the contraction, and basically, I hope you can see it here in the upper right-hand corner, also in the shape of the contraction curve. And this basically has something to do with the modulation of the calcium influx, which is, you see here the colorful green line, that's the normal, and the red line, that's the lipofuscin-loaded cardiomyocyte, and you see the calcium kinetics in these cells is different. We do not know yet why this happens, why is lipofuscin influencing the calcium metabolism in cardiomyocytes. But what I can tell you, it's the same in beta cells, the insulin-producing cells. Also there, lipofuscin has an influence on the calcium metabolism. So, yeah, it's basically in one of the functions, so it can impair the cell function. And as you might realize, cardiomyocytes and beta cells, which are important for diabetes, it's really important for disease development, or major disease development, cardiovascular disease and diabetes. And then we did an experiment with Johannes Trougakis from Athens, and I want to focus your attention on the Drosophila experiments here, where we fed Drosophila with lipofuscin in their chow, and you can see that the life expectancies of this Drosophila is tremendously declined compared to the control. We could show that this is a really going of lipofuscin into the animal, into the Drosophila, accumulating in lysosomes there and influencing the metabolism of various cells. So that's basically the functions of these protein aggregates. And now the question, we should get rid of it. So if it's so detrimental to many of our cellular functions and bad, we should get rid of it. And we did some experiments at the very beginning, and we tried a different set of proteases here. As the protease K, you can see it's the first column control, then bromelain, different other plant and microbiota derived proteases, which are very, very active, and used or very often to degrade everything, every protein which is available, and the combination of them, but still there is a lipofuscin. It's still only degrading the loose peptides I showed you and not the core of the material. And then we use the core of the material and tried whether some bacteria can degrade it. We isolated a population of bacteria from soil, and as you can see here in these two Eppendorf tubes, you can see after some time the lipofuscin volume is reduced, and some of these bacteria are growing pretty well on this lipofuscin as the only carbon and nutrient source at all. So at this point we stopped and we applied for the SENS Foundation, because basically we are not interested, or no one is really interested in the degradation of artificial lipofuscin, but real lipofuscin is important here for us. So we got a study running now, so besides the other project with the SENS Foundation, of the characterization of natural lipofuscin and finding ways to destruct this. Annika Höhn, she did a lot of these experiments to the effects of lipofuscin, is coordinating it, and Tim Weidensberger is the actually postdoc working on it. All my co-workers here, Annika Höhn and Tim Waldensberger, these are the two people working on it. And thank you for your attention, and I'm happy to take your questions.